Today I am recording this video for Biosciences 423 at Simon Fraser University. It is 2021 spring semester. It's springtime and it's beautiful out. And I'm going to be talking about my research. All right, goals for this lecture. I would like to give you, the student, a bit of a taste of graduate level research. Because this is a 400-year course, 400 level course, 400 years, sometimes it feels like that, a 400 level course, I'm presuming that you are curious about what it's like to be a graduate student and to study research. And to be in academia as a lifestyle, doing bench work. So this will kind of get into the nitty-gritty of how one thinks as a scientist. This is the moment in our course where we're transitioning from talking about the developmental nitty-gritty and the genes regulating the development, and we're bringing that forward towards how it affects function and behavior and movement. So this lecture is going to bridge that. But really what's going on is that I like to talk about myself and I like to talk about my research. So this is me wanting to share the things that I'm excited about with you. Important information, blue text in this color is take-home message. So if you see this blue text, it means I want you to remember that thing. Black bolded text like this are going to be vocab words, and there will be a handy dandy list at the end. I have not put these notes up online yet. When I do, that vocab list at the end will be the correct vocab list for words that you are expected to know the meaning of. And orange text are questions that I want you to think about. Either they will be multiple choice questions in the lecture, or they'll just be questions I had to think about as a scientist. Okay, so let's get going. Context. This is review. Early growth cones are guided by environmental stimulus. Later axons follow those earlier pioneering axons, and this is a highly conserved process. We have seen this before. This is not news. This is also not news. I think you've seen this exact image before in this course. But I do want to review it real quickly because it's important. Specifically, AVG, this cell body here, you can see my mouse, extends its axon along the right ventral nerve cord. And then later, here are our followers. We say our interneurons here in green in this instance, though that's not going to be consistent, and I'll tell you when it changes. And we see our motor neurons in red and blue and purple, which are different types of motor neurons. And again, we'll get back to the color stuff later. But for now, I would like to show you the extension of the AVG axon in real time. So this is a video of an egg of a C. elegans developing. And in this video, there's a fluorescent protein associated with neural identity, so neural genes. So you'll see neurons kind of attaining their neural fate as you go. So here we go, and that is the head. The head. And you can see the head growing, and that. Okay, so here's our head. And we can see here's our ventral nerve cord, and here's that first cell to extend down the ventral nerve cord. It's our AVG. And as it goes, watch around it, you're going to see more cells gaining their identity. And now that it started twitching, you can tell that there are more cell bodies in this area. Those are motor neurons. It's twitching because now there are neurons innervating muscles in that region. And you can also see that this axon is getting brighter because more axons are following it. So there are multiple axons. You can see the growth cone at the end too, which is kind of ridiculously cool. Look at that. Ah! Ah! And there it goes flailing around. So that's a baby C. elegans that's extending right now. You can see its ventral nerve cord is now quite large, and it has a bunch of neurons all along it. Those are all the motor neuron cell bodies. There it goes. Okay, so I study the pioneer-follower relationship of just the right ventral nerve cord. And here's a new image with all of the neurons together. Here's our AVG now in green, and it will forever, always going forward be in green. And you can see the red is going to be command interneurons. Command interneuron axons are kind of organize the motor system. Those are these red things here. This is not something that I want you to memorize. Please do not memorize this thing. These are all of the different cells that organize the motor system and the motor response system. And that is all happening here. So all of these synapses, these connections that you're seeing between the sensory neurons, the red command interneurons, and our purple motor neurons, which is also going to be consistent. All of those synapses, all of these arrows and lines happen en passant, so in passing. What that means is here's our C. elegans, there's its brain, and all of these little bright things, these are all locations where synapses are happening in passing. And here we can see, here's a DD motor neuron, and here's RID, which is one of those red interneurons. And you can see the way that their synapses work. This is an electron micrograph image, where this is the ventral nerve cord. You can see all these bumps are synapses. So each interneuron axon must be in the right place in order to make the correct synapses. Each motor neuron axon must be in the right place in order to make the correct synapses. So the organization of the ventral nerve cord is vital. And if it gets messed up, and it can be messed up. Okay, so Richard Durbin, an exciting name that you don't need to memorize, but but he was the person who took electron micrographs of sections of worms down the length of it and completely reconstructed the nervous system. He also ablated AVG, so went in with a little laser and killed the AVG cell. And he found that after 3D reconstructing those specific worms after they grew up, when you kill the AVG cell before it can extend its axon, those 3D reconstructions of the ventral nerve cord that he did showed a very disorganized ventral nerve cord. Those are ugly pictures. These are pretty pictures. Dr. Hutter, our professor, was able to do very similar experiments, but this time he used fluorescent markers. And in this specific image, we're seeing motor neurons in red and blue, and we're seeing interneurons in green, but that will change. In the future, interneurons will be red, motor neurons will be purple. So don't forget about that. All right, so here we have, a fun multiple choice question, because everything's more fun with multiple choice questions, right? Right? Okay. To find a gene involved in AVG axon guidance, we, A, remove genes for likely guidance cues and look for AVG defects. B, remove genes known to affect command interneuron axon guidance and look for AVG axon guidance defects. C, cause a bunch of random mutations and look among those for AVG axon guidance defects. Or D, any or all of them would work. So take a moment, pause, think about it. But all of these would work.
Oh, interesting.
Mm-hmm. And usually you do all of them in order from easiest to hardest. And we tried in our lab to do them all. We started from the top. Because this is how science works. You throw everything at the wall and see what sticks. So how does AVG find its way? Well, let's start from the top. Pick some genes that are important for axon guidance, knock them out, see if it messes up AVG axon guidance. So we did that and none of them worked. So now what? Now we go back. So the next one, specifically look at things that affect those follower axons, right? There are a lot of those. There are hundreds and hundreds of them, like so many of them. There are so many of them. So we looked at that option and we decided that it was easier to jump to C and do a genetic screen. So I have been hammering this through your brains from the very beginning of this course, the importance of understanding how genetic screens work in this class. It's important because it's a lot of what Dr. Hutter does in lab. And these are the steps. You randomly destroy genes. You find the mutations that cause defects. You sequence and locate the gene. And then you try to figure out what its function is. So let's start from the top. This is what we looked for. These are the phenotypes. Green is AVG. And here are various ways AVG can mess up. This is the one that we ended up focusing on. But in the first genetic screen, this is the one that we uncovered. There was only one mutation in that first genetic screen. Usually there are like 20 or 30. So it was weird to find only one. Interestingly, that one ends up helping AVG orient itself along the anterior-posterior axis by way of that Wundt gradient that we learned about all those months ago. Again, fun thing for your knowledge, don't memorize it. But we were expecting to find more. We didn't because of redundancy. So redundancy, the concept of redundancy is basically that different factors will all do basically the same thing so that if one fails, others will pick up the slack. And the analogy I want you to think of is a spider leg. A normal spider has 8 legs. However, it's okay with 5 legs. It's even okay with 4. All of a sudden, if it only has 3 left, it can barely walk. And if it only has 2, it's dead. So in order to identify any of these legs, we need to start by taking out more than one. And then the effects of the remaining legs can be observed. Can you tell where this is going? It's going to an enhancement screen, I'll tell you. But this was the idea of the many legs. Nitogen. Nitogen is part of the basal lamina. If you remember, the basal lamina and laminin as part of the basal lamina, and netrin as sticking to laminin in the basal lamina as these ringing cymbals, because the basal lamina is a substrate for axons to extend along. That's what we're talking about. And nitogen is one of those things that incorporates itself into the basal lamina and helps it be a substrate for axons to extend along. So the theory is that if you remove nitogen, there will be more difficulties that are completely unrelated to the axon guidance factors that we're trying to see the function of. So taking out some legs. Luckily, without mitogen, mice are very sick. Humans are stillborn, but C. elegans are fine, except they have midline crossing defects at about 12%. So that's convenient. So nitogen, perhaps, is a substrate to which several legs are attached. Without it, we may see more clearly the effects of disrupting the remaining legs. So how do we do this? We can look directly at nitogen mutants and see if they have extra defects. We can run a genetic screen in nitogen mutants, or we can add candidate genes to the nitogen mutants and try to prevent defects. Again, I want you to sit down and think about this and make sure that you understand the correct answer. Yeah, so the reason why it is an A is we already looked at A. There's 12% ABG defects in nitrogen mutants. We know the answer to this question. We don't need to look anymore just at nitrogen mutants. And we've already talked about how we tried all the candidate genes. There weren't any good ones. So this is the last option, is running that genetic screen. And it's specifically an enhancement screen where you start with a mild phenotype and you're trying to make it worse. Does that make sense? So when you mutagenize our nitrogen 1 null worms, or when we mutagenized our nitrogen 1 null worms, we got this wide array of defective worms with fairly intense ABG defects. So this is great news. It means that we successfully removed enough legs to be able to see the loss of the remaining legs. And if that phrase confuses you, pause, rewind, and try to work through the role that nitrogen is playing in this process, because it is an important process. When we remove nitrogen, we are disabling the worm so that we can see its coping mechanisms, so that we can see the other ways in which the worm, in which the ABG axon is finding its path that are unrelated to nitrogen. Okay, so next step. These are just letters and numbers. These aren't actually gene names because we don't know which genes we mutated in our screen. So what are these genes? Each strain has many mutations. Only one causes the phenotype. So we're going to sequence and map. We're going to confirm allele identity and gene function. And we're going to identify our biological mechanism. And we're going to do all that in 333 and 202. And for this lecture, I'm going to skip all that and get right to the fun stuff. This is the fun stuff. We're all disappointed, I know. So two of the genes, the mutations that we identified, were a mutation in AEX3 and a mutation in CCD5. And both of these were associated with the same pathway, which is a pathway that regulates embryonic vesicle transport. So these are the vesicles that are holding the stuff that the growth cone needs and wants to use. And these genes, CCD5 and AEX3, are proteins that the genes code for proteins that are involved in transporting the vesicle that holds the things down the axon to the growth cone so that it can do stuff. And specifically, what it's transporting has something to do with UNC5. This is the information we have. The information we have is that these two genes share a pathway with all of these other genes. And we know a fair amount about what those other genes do. So based on this information, that AEX3 and CCD5 play a role in transport, and that transport is linked to UNC5 function. Why do AEX3 and CCD5 mutants have misguided axons? Here's another fun question. Fun questions. So pay attention. A, the growth cones can't recognize netrin anymore. Reminder, these UNC5 is that netrin receptor with UNC40. So A, the growth cones can't recognize netrin anymore. B, the growth cones now respond incorrectly to netrin. So they can recognize netrin, but they can't respond correctly anymore. C, transport to the growth cone is messed up generally, which can cause any number of problems with axon guidance, and we have no idea which ones. Or D, we actually don't have enough information to draw any meaningful conclusions, hashtag science. What do you think is the answer? You think it's C. I also think it's C, but really, that's just a hunch. I was discussing this question with Dr. Hutter, and we both agreed that it made the most sense to say that the answer is D. And Dr. Hutter emphasized that this is one of the take-home lessons of the entire course, that so much is left unknown, and so much of research ends in having contributed some information, but not all of the information, and most notably not the information you wanted. However, this does take me to the end of my ability to study the mechanism of CCD5 and AEX3. So you need 110% of the answer to make a conclusion. You need to conclusively prove that none of the other options are true. Remember the HA and HO, you have to disprove your HO in order to prove your HA. Going all the way back to intro bio. Science, science. Yeah, so at the end of every paper, everyone says, but it could be something else. We're not entirely sure. We look forward to other people exploring these concepts more, but I'm done now, bye. And this is the moment where I get to say, but I'm done now, bye, because my research into the actual mechanism of CCD5 and AEX3 ends with this. And in this moment, this is where we're going to cut for the video number 1, and we'll move on to video number 2, enthusiasm.